wabarakatuh. First of all, pertama-tama marilah kita bersyukur, memanjatkan puji dan syukur kita kehadiran Tuhan yang Maha Kuasa Allah Subhanahu wa Ta'ala, karena berkat limpahan nikmat, sehat, dan juga kesempatan yang masih diberikan kepada kita sampai dengan hari ini, hari Jumat yang cerah. Alhamdulillah, setelah kemarin siang, sore, dan tadi malam hujan badai, saya lihat di beberapa wilayah, Bantul, Kota, Sleman. Tapi pagi ini alhamdulillah Jumat berkah ya, mudah-mudahan Bapak Ibu dan saudara-saudara sekalian, sebagaimana yang menjadi agenda kami di jurusan Pendidikan Biologi, khususnya Prodi Pendidikan Biologi, hari ini kita punya acara yang luar biasa, alhamdulillah, yaitu sebagaimana yang kami sampaikan, kita akan ada talkshow, diskusi bersama ini rekan saya, kolega saya. Jadi ceritanya, saya bertemu pertama kali dengan Pak Rashid for the first time. I knew his names kebetulan saya ketika studi di Australia itu saya bekerja paruh waktu sebagai cleaning service di sekolah beliau. Jadi tempat kerja saya itu di Rossmoyne Senior High School, dan when I just doing my duty, ketika saya bekerja, saya melihat-lihat, biasa karena di kantor juga di lab begitu, saya tertarik ada nama Rashid Rafey dan he's doing biology teaching.
Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh.
Assalamualaikum warahmatullahi wabarakatuh.
Selamat pagi, salam sehat dan semangat untuk Bapak Ibu semua. Perkenalkan, saya Kartika Lama Pertiwi, saya dokter. Saya adalah dosen pada jurusan Pendidikan Biologi FMIPA UNY. Saya adiknya Pak Agung, jadi saya masuk di sini 2006. Atas nama Fakultas Matematika dan Ilmu Pengetahuan Alam, kami mengapresiasi, berterima kasih atas kesediaan dan kehadiran Bapak Ibu pada acara di pagi hari ini. Mr. Rashid, on behalf of OLD, the Faculty of Mathematics and Natural Science, I would like to warmly welcome you to our faculty, and thank you for your kindness and willingness to be our speaker today. Saya ucapkan terima kasih kepada kolega saya, Bapak Agung Jaya PhD, dan juga timnya, dan dosen-dosen muda kami yang telah sukses mengeksekusi acara pada pagi hari ini.
Kita akan langsung saja menyimak paparan dari Pak Rashid. Silakan, please.
We'll try to mention two topics, as Pak Agung said. I will be doing it in opposite sequence. I will talk about science and human endeavor in educational curriculum and how it empowers our students to learn about society and how science impacts society. Later, I will go into the challenges in teaching biology, contemporary biology. Okay? And I will share with you some ideas and strategies I use in my classroom on how to teach contemporary biology. And I'm referring to year 12 human biology course. Okay. Jazakallah khair. together around each table. Could I have 6 willing groups who would like to practice an activity on epigenetics? Yeah. Don't be shy. Yeah. I'll take you through it, so it should be okay. The first thing it tells like, yeah. Yeah. Especially when you are dealing with food, if you give it to the kids and you turn your back, they will eat it in 2 minutes. Okay? Then you come back, you want to do something, it's all gone. So the first thing you do is tell them what you are about to do and demonstrate before you give it to them. If you give them the food, that's it, it's all gone. They'll be eating while you're talking. So just show them exactly what you need them to do so they can do it. And explain very slowly. So tell them, I'm going to use this piece of licorice as a DNA. This is my DNA strand. And here is what they call the nucleosome. This is a protein that DNA wraps around. Okay? And that's up. So use a toothpick to hold them together so they don't fall apart. So you got 2 and 2 and 2 and 2 becomes 8. So you get an octamer. You got 8 histones in the nucleosome. Okay? Now, this will be the same. You put 4 toothpicks in. And then you want to show how epigenetic works. So you'll put the DNA, wrap it around the nucleosome. Then on here will be another nucleosome. Lots of nucleosomes around the DNA. And it goes something like that. So the kids would look at a section of the DNA with nucleosomes. Like that. So now I give them also a berries. Thank you, I can see. So now I'm going to show them 2 different processes. Acetylation and methylation. Acetylation is adding an acetyl group to the tail of an histone, or an amino acid of a histone. So this is an histone protein. So I would say, let's add an acetyl group to a carbon molecule. And I'll put that in. So that histone has been acetylated. What would be the impact of acetylating a histone? It will open up, relaxes the bonding between the bases that makes up the DNA strand. So the moment you acetylate it, it will open up. Then, only then, when DNA opens up, you can now copy part of that DNA or a gene. That gene now can be transcribed or read by messenger RNA and then can be translated in a nucleus on a ribosome. Okay? So if it is unacetylated, what happens? That will induce bonding between the bases on a DNA strand, and that will cause it to wrap tightly. So now my gene, part of the DNA, is not exposed. I cannot read that gene. I cannot read it and translate it and make more proteins, because actual protein itself, the DNA polymerase need to copy it, and it cannot have access to that gene if it is tightly coiled. So use acetyl group to open it, and then you can copy. Once you copied it, if the body doesn't need it anymore, it will take off the acetyl group, allows it to bond again together. So now the gene is turned off or switched off. You cannot copy it. And some diseases are due to genes being switched off. If the gene is switched on, we can make the protein. Like, for example, cystic fibrosis. The disease affects the lungs and pancreas as well. The gene is switched off, so it doesn't work properly. It doesn't make the correct protein. Because it doesn't make the correct protein, our lungs don't work well. We end up having water accumulating in our airways, and we actually can drown on fluid in our lungs. And we cannot make the enzymes in our pancreas go to our small intestine, so we cannot digest food. So that's acetylation, or sometimes I will say, what about methylation of DNA? So if you get a methyl group, and I will use a different color, and I tell them this is a methyl group now, not an acetyl group, so that's red, mera, is a methyl group. If you methylate a cytosine onto DNA, and not any cytosine base, it must be a cytosine adjacent to guanine. We call it CPG, cytosine phosphate guanine. Only that cytosine, which is close to guanine, can be methylated. And if that happens, and that will prevent that gene from being transcribed and translated. So it will be turned off or switched off. That protein made by the gene cannot be produced anymore. And that could have a bad effect or good effect. You never know. Some genes, actually, when they are turned off, they prevent problems, like cancer, for example. When they're turned on, they can cause some diseases. Okay? And a lot of people nowadays, they ask you to eat your veggies. If you eat lots of green vegetables, like spinach, kale, Brussels sprouts, they have lots of methyl groups in that food. So the more you methylate, the more likely to be able to produce protein or transcribe and translate protein from your DNA. So you're less likely to become sick. And that's what he tells the kids. If you got very scientific kids, you tell them why you should eat your veggies using scientific evidence, okay, in such a good way. So that's what I get my kids to do in order to understand the process of methylation and acetylation and how impact on DNA, whether it could be transcribed and translated or not, whether I can make a protein or not, and that impact on me, whether I am healthy or sick. It's based on that process of acetylation and acylation. So this is one thing the kids will use, and it makes that topic of epigenetics very important, because I would say, well, my genes hasn't changed. No, they haven't changed. Exactly the same sequence of bases. The only thing I've done is I attached an acetyl group or a methyl group on your DNA. I haven't changed your DNA at all, and that's why we call it epigenetic, above genetic. It's got nothing to do with the gene. The genes are still the same. I've just added something that stops your genes from being read. That's all. So the body can take it off and can add it on as well. So it depends what we have in our body, what we eat, and that will affect methylation or acetylation. Okay? So if you eat, for example, your green veggies every day, you get a lot of methylation happening on your DNA, and that prevents most diseases from taking place. Okay? You look at Australian people, they don't have a lot of veggies. Okay? They don't eat much veggies. Every time I go to the market, I see a large population of Chinese going to the market buying raw vegetables and fruits. Aussies, they go to a restaurant and they order chips, fish and chips, McDonald's, junk food, and Diet Coke. Done for Diet Coke. And that's why they get sick, because there's sugars and lots of additives in food. So to make sure you get healthy, you're gonna make sure how you eat and explain to the kids how healthy food can impact on your DNA and how it works. Can make you feel sick or not. Okay? Jazakallah khairan. Terima kasih.
Saya rasa Pak Rashid sangat gamblang untuk bisa mencontohkan kepada kita bagaimana epigenetik. Saya tidak tahu, di sekolah sudah sampai pada epigenetik mestinya ya. Jadi bagaimana mekanisme gen itu akan aktif atau tidak, switch on or switch off. Aktif tidaknya sequence gen di dalam DNA, pada DNA itu kan akan memberi implikasi terhadap muncul tidaknya kelainan, penyakit, atau kita bisa sehat atau tidak. Kalau tadi misalnya di sini Pak Rashid mencontohkan misalnya pada mekanisme acetyl dan
methylation and acetylation.
Acetylation dan methylation. Acetylation itu terjadi karena apa? Karena ada unsur asam di sana yang menyebabkan terbukanya ikatan histon pada DNA kita, kemudian memungkinkan kalau dia terbuka berarti ada sekuen gens yang ada pada ikatan histon itu yang mungkin untuk mengalami transkripsi dan sehingga nanti menghasilkan protein. Kalau sekiranya protein itu ternyata yang tidak baik, maka kita akan menjadi sakit. Begitu juga pada methylation tadi, methylation khususnya pada ikatan C dan G, guanin dan sitosin, fosfat guanin dan sitosin. Methylation itu banyak dikandung pada sayuran. Itu sebabnya tadi beliau berpesan kepada kita, kalau kita mau menyarankan atau meminta siswa kita makan sayur, itu secara ilmiah bisa dijelaskan dengan epigenetik, bahwa sayuran hijau itu banyak mengandung kandungan yang memungkinkan terjadinya methylation, sehingga protein-protein yang ada pada ikatan pada sequence ya, dari gens di sequence yang mengalami methylation itu memungkinkan si gen ini untuk aktif dan membatasi munculnya penyakit-penyakit dari protein tersebut. Dan ini penjelasan ilmiah mengapa kok makan sayur itu penting. Bukan semata makan sayur itu penting karena mengandung serat sebenarnya, tetapi bagaimana serat itu bekerja pada ikatan DNA, CG, pada sekuensi gens yang memungkinkan kita tetap bisa sehat karena protein dari CG tersebut yang aktif. That's all. Bapak Ibu sudah membaca worksheet-nya ya? Oke. Pak Rashid menyediakan secara khusus marshmallow dan juga berries ini. Silakan kalau misalnya ingin mencoba mengambil berries-nya ini.
Your explanation, I can assume that your biology class, it must be very attractive, it must be very interesting for students to learn. But I'm very curious to ask about the teaching and learning process in Australia, in your classroom or in Australia as a whole. Is there old-fashioned way of traditional teaching like direct instruction is still used in your classroom? Direct instruction is teaching directly to the student, transferring the knowledge, transferring the information, and so on and so on. Thank you.
Thank you. Yes, of direct instruction, but we don't use it predominantly all the time. Okay? As a teacher, you have to talk to the students, you have to make sure they understand by giving direct instruction, but not the whole period, like a lecturer at university. You walk into a lecture theater with 400 people looking at you, and you talk for 2 hours, and then you go. We don't do that, okay? But we give some instructions for our students in order to learn what they need to learn, and then having listened to us, giving direct instructions, now they have to learn on their own. So called the student-led instruction. So now they have to go and read what I've just talked about and research it. And they come back to me and they will tell me what they've learned. So I will introduce a subject. I might not actually tell them exactly how a certain concept works. I will say, well, today we're going to look at epigenetics. Epigenetics is something that works beyond genes. So it has to do with chemicals impacting on genes, whether they are expressed or not expressed. Therefore, they will promote or inhibit protein from being made. That's all I say. Just give me the definition of epigenetics. You tell me how is a gene is turned on by doing research. So I give instructions, I tell them what to do.
then expect them to go and research it. So for young kids, you want to do that with them. Like when I teach year 8 or up to year 10, lower school, I ask them to do a lot of research to get them active, because the young kids will not learn from you the whole time. They want to be active. You have to make them active learners, responsible. So they will have to actually actively go on the computer and research. If you expect them to sit there and look at you and listen the whole period, you're mistaken. They're not going to listen. They will lose them. 5 minutes after that, they're gone. They'll be playing games on the computers and you don't know. So give direct instructions. That's important for students so they don't waste their time. When you are very clear in your instruction, direct instruction, then the kids can do their job, their task efficiently. And then you stop them and you see how they went. And then you give them more time. And then you interrupt. Okay, what happened next? Get some feedback from them. Feedback is very important. Okay. But direct instruction is important part of teaching, but doesn't take the whole period. Yeah.
So, Bapak Ibu, apa yang tadi dibagikan oleh Pak Rashid, setidaknya ada dua hal penting yang bisa saya coba wrap up, saya coba menutup lah ya. Hal-hal mendasar sebenarnya saya yakin kita semua sudah memahami. What is method, what is strategy, metode, strategi media, apa yang tadi dicontohkan oleh Pak Rashid, kita semua sudah tahu itu. But encouragement, di sini yang saya lihat mungkin apa yang bisa menjadi penting, bagaimana kita berusaha menanamkan betul pada diri kita, keyakinan kita, teaching belief kita bahwa we need to do the best for our students. That's the point. We need to do the best for our students. Meski ada faktor lain yang kedua, tadi yang juga sudah disampaikan oleh Bu Dr. Tiwi di awal, ada beberapa faktor mungkin yang memang jelas membedakan antara kultur Indonesia dan Australia, kurikulumnya jelas berbeda dan sebagainya. Tetapi hal pertama tadi bahwa sekiranya kita bisa menanamkan atau kita mencoba, kita terus bertumbuh untuk bisa menjadi inspirational teacher, saya yakin hal-hal yang tadi dicontohkan oleh Pak Rashid bisa kita coba adopsi dan kita terapkan. Jadi tadi saya rasa apa yang disampaikan terakhir tadi, menurut saya ini sebuah hal yang penting. Thank you for being a teacher. Yes. Terima kasih.
Bagiku ya, ini untuk pertama kalinya aku belajar biologi dengan objeknya itu pakai makanan kayak gitu.
Jadi untuk diterapkan ke siswa itu menurut aku kayak lebih menarik aja. Karena kan pelajaran epigenik itu kan cuma awang-awang aja, sedangkan ini direalisasikan menggunakan makanan, jadi lebih menarik untuk siswa untuk belajar biologi.
Ya, selain itu juga siswa akan lebih bisa memahami sih, karena konsepnya ini kan abstrak gitu. Oke, terima kasih.
Kesan saya selama mengikuti acara seminar ini adalah sangat mengesankan ya, karena kita bisa melihat berbagai pandangan mengenai cara mengajar biologi, tidak hanya di Indonesia saja tetapi dari Australia seperti itu. Sangat terkesan dan sangat menginspirasi, terutama untuk calon guru hebat di masa depan nanti. Amin. Terima kasih.
Acara hari ini luar biasa, sangat menginspirasi, menambah pengalaman, menambah motivasi juga. karena kita bisa mengetahui pembelajaran biologi di Australia yang tentu ada perbedaan dengan di Indonesia begitu. Terima kasih, semoga ada kegiatan semacam ini yang lain kali lebih sering lagi begitu. Terima kasih.